Okay, so it's 4:15, so welcome to the next session. Hope you had a good break and a drink, cup of tea, whatever. We've got two great sessions left, one on inflammation and then a final plenary from Gary Rosenberg.
Sorry, this isn't the one I just sent, actually. Have you got the one I sent? If not, I'll send it myself. This is a previous version.
Okay, have we got Hugh's latest, or maybe he can screen share?
Screen share it.
Just while you're sorting that out, this session is on inflammation. So clearly neuroinflammation is really a big topical area in degenerative dementia, where there's increasing evidence that it's a drive or an accelerator of pathological change. There's clearly a large literature in terms of after stroke. But I think what's less clear and what this symposium will tackle is its association impact on small vessel disease. And my co-chair is Frank Eric. I don't know if you want to say hello, Frank Eric, or wave. And we're going to alternate introductions between us. We'll have all the discussion, I think, at the end, unless we have time unallocated during each talk. So it's a pleasure to welcome Hugh, who I hope is able to have his right set of slides. Hugh's well known, I think, to everyone here, co-organizer of this meeting, along with myself and Raj Kalaria, professor of stroke medicine at the University of Cambridge, previously at St George's. Well known for his work on genetics of small vessel disease and on CADASIL, and also on vascular cognitive impairment, and also the associations of non-cognitive symptoms like apathy and depression, and also involved in very important work on neuroinflammation, which, Hugh, you're going to share with us today, talking about PET studies of microglial activation and blood-brain barrier dysfunction in small vessel disease. And you're ready to go, which is great.
Can you see it now, John?
Yeah.
I've just been able to share now, so that's great. Somebody's authorized it. Perfect.
Just needs to be put on full screen mode.
That's great. Thank you so much, and it's a great pleasure to speak in this inflammation session, which I think is an area that's becoming quite exciting and interesting. And in many areas of neurology and psychiatry, there's a lot of evidence that inflammation may play a role. Whether it's actually causal or not, we don't know.
To quit.
So I'm talking about cerebral small vessel disease. I don't need to say anything more about this because we've heard a lot about it already. But conventionally, when we think about these white matter lesions that are such a hallmark of the disease, we thought of them as being due to hypoperfusion and a small vessel arteriopathy and chronic hypoperfusion and ischemic damage. But evidence over the last 10 to 20 years has suggested there are many other processes involved here, one of which appears to be blood-brain barrier permeability, and an additional sort of more recent risk factor may be chronic inflammation. And of course, this could be inflammation either from the systemic circulation or from the brain itself. And the two of those, increasing evidence suggests the two of those can interact in a sort of two-way process with inflammation systemically driving inflammation within the brain itself, as well as inflammation arising within the brain itself. Now, there's a lot of data on systemic inflammation in small vessel disease, and this is a very nice paper that Audrey Lowe carried out with John and myself, sort of summarizing all this data. So an interesting read if you're interested in the area. But I'm not going to talk about this very much because we've got a really exciting talk following mine, talking about the evidence that immune reprogramming in the peripheral blood may actually play a role with in small vessel disease. But I've just summarized some of the old data here to suggest that systemic inflammation may relate to small vessel disease. So there's, for example, a large number of studies suggesting that markers of inflammation and endothelial activation associate with SVD in cross-sectional studies. So IL-6 and CRP in this study from the Charge Consortium, that they may predict small vessel disease prospectively. So important if we're thinking of whether they're causal. And this is data on ICAM1 and IL-6. And then another way you can look at causality is to do Mendelian randomization and select out genetic variants that predispose throughout the lifespan to increased activity in a particular pathway. And there's evidence from this as well. And there's a nice review again summarizing all this data if you want to look at this by Pogesi et al. But what about inflammation in the brain itself as opposed to peripherally? And this, of course, is much harder to look at. There's some post-mortem data. This is data from the Sheffield group showing microglial evidence of activated microglia in the brain associated with small vessel disease. Of course, these are all post-mortem studies, so they're right at the end of the disease spectrum, and you're always a bit worried about what post-mortem damage could have done. There's also evidence from animal models, though, of small vessel disease that inflammation may play a role. But there's very little evidence in man, in vivo. Now, we really got interested in this area when we read this paper by Gary Rosenberg, who's speaking also in this session, who created a very elegant model of small vessel disease, a rodent model, where chronic hypoperfusion was induced. And this was followed by a whole cascade of events with hypoxia, an inflammatory response, matrometallic proteinases production, blood-brain barrier breakdown, tissue damage, and a sort of vicious circle where tissue damage can increase the inflammatory response. And after talking to him, we wondered if we could sort of investigate whether a similar cascade happened in man. So working with Gary, we tried to implement similar techniques, but in man instead of a rodent. Now, Gary looked histologically for inflammation, but we obviously can't do that in the brain. So we've been looking with PET. So we use a ligand called PK195, which binds to activated microglia. So it gives us an idea of if there's activated microglia there. And at the same time, using a PET MR scanner, we carry out MR while we're carrying out the PET, and we measure blood-brain barrier breakdown. So this is using a permeability technique that many people have used. We used the one which Gary had actually developed and used in his patients and in his models, which uses a lower dose of gadolinium and allows you, using PATHLAC modeling, to get a map of blood-brain barrier breakdown within the brain. So you can look at the spatial pattern of blood-brain barrier breakdown. So these are the techniques we've done, quite time-consuming, you can see, and quite expensive. Now, in our initial study, we recruited 20 patients with sporadic small vessel disease, so all lacunar infarcts with some white matter hyperintensities. We compared them with 20 cadacils, all mutation positive, and 20 healthy controls of similar age. And we just did the standard assessments, cognitive testing, samples for cytokine assays, which I'll come on to, and then the imaging I've told you about. And this is work that Jess Walsh carried out for a PhD. And this shows the sort of pattern, the images that can be derived from these scans. So what we've looked at here is hotspots of increased permeability and increased ligand uptake above a certain threshold. And you can see that the white is areas in an individual patient where there's increased microglial activation, and the green is areas where there's increased BBB permeability. So these are individual patient slices. There. So you can really get quite nice images using this. So here's a quantitative analysis. This is initially the PK119 binding. So this is the microglial activation. And you can see that these are the normals. Here's the sporadic SVD, here's the cadacils. And you can see that the mean binding is increased in the cases in the normal appearing white matter. You have to be a bit wary about this because there was so little lesion in the normals. So this is a very wide confidence intervals. And we both looked at mean binding over the whole of the region, but also the volume of these hotspots above a certain threshold. And you can see these are also increased. So it does look as if there's increased microglial activation in the white matter, both in sporadic disease and in cadacil. This is the blood-brain barrier permeability using the gadolinium contrast. And you can see we've just presented in exactly the same way, both with mean permeability and also the volume of hotspots. And you can see here that the permeability is markedly increased in small vessel disease. This has been shown by many people, initially by Joanna Wardlaw and colleagues. But there's not such an increase in the CADASIL cases, much less, which is interesting, and that wasn't significant. So the next question is, we've shown these two processes are occurring, but how do they relate to each other? Does this blood-brain barrier breakdown, which can occur sometimes in these cases around the edge of the white matter lesions? These green images are just white matter lesions which have been drawn around, and the blue are white matter lesions which have been drawn around here. This is another patient with the glial activation. Are they spatially related? So we did a voxel by voxel analysis to see whether they were more likely to be related or not likely to be related, and we found there was no increase in overlap between the voxels, so suggesting that there was no spatial relationship between the two processes. So at least it doesn't mean they're not related over time, but at this individual time point, they weren't related spatially. We also tried to see if there was any change in these characteristics in the penumbra around the white matter lesions. So we expanded the white matter lesions out 3 millimeters, and we looked at BBB permeability and binding within this sort of penumbral region, just in the 3 millimeters around each white matter lesion. And in fact, we found no difference in permeability. So there was no suggestion that there was increased permeability adjacent to the lesions. And we actually found a reduction in microglial binding around the white matter lesion. So this might mean that perhaps the tissue is already damaged to a degree. We know it's a bit abnormal on DTI. So we also looked at biomarkers. So we had one of these Olink panels of cardiovascular markers. We looked at 92 markers. We added in CRP. Because you have so many markers, we used principal component analysis and selected out the first principal component to sort of get a measure of the variation in these markers that we could correlate with our imaging. And this first component accounted for 33% and 30% of the variance in sporadic and CADASIL respectively. And we found that the biomarkers correlated with BBB permeability in sporadic SVD, but not with neuroinflammation. So it looked as if there was some relationship between this systemic cytokine or activation and BBB permeability, but as I say, not with CNS inflammation. So in this cross-sectional study, we showed in sporadic small vessel disease, both microglial activation and BBB permeability had increased. We showed microglial activation had increased in CADASIL, but not convincingly BBB permeability, and no spatial overlap. Now, there is a little more data to suggest there is increased PK111 binding in small vessel disease. This is a study from Audrey Lowe in actually a cohort of patients who had MCI and Alzheimer's disease. But what she did was she quantified the small vessel disease in them and showed that the amount of whole brain ligand binding related to the amount of small vessel disease. So there is some supporting evidence from other studies of this relationship between neuroinflammation and small vessel disease and white matter hyperintensities. But of course, all we've shown is there's an association, and this could all be secondary or it could be primary. And to address this, we need to do longitudinal intervention studies. So what we're doing is looking at 1 year follow-up in these patients, and Dan Tozer is going to present a little of this data in a moment, so I won't deal with it further. But essentially, we're following all these patients up to a year where we're doing DTI to look at progression of white matter damage, and we're hoping to follow up in the longer term as well to clinical cognitive decline. But what about an intervention study? And the bit I didn't show you about Gary's work was the intervention. So what they did was show this cascade, and then they sort of hypothesized that minocycline, which has anti-inflammatory effects and also sort of stabilizes the blood-brain barrier against MMP damage, might be a possible treatment. And indeed, they showed that in these rat models that if you gave minocycline, the damage was significantly reduced. So here's some white matter damage with vehicle, and here it's been markedly reduced with minocycline, and you can see this is shown quantitatively here. So what we've been doing is exactly the same, trying to translate this animal work into man. So we've, in the second part of our study, we're doing the Minerva study where we're giving a double-blind randomized controlled trial of minocycline versus placebo. And our readout is blood-brain barrier permeability and microglial activation using MRI and PET, with secondary markers of biomarkers in the blood of inflammation and endothelial function. So this is our design. So we randomize patients who have all got small vessel disease and some white matter hyperintensities. We then do a baseline PET MR, give them minocycline or placebo, re-image them at 3 months, which is our primary outcome, and then we continue to a year, just also with DTI as well, and MRI, but without the PET MR. And we need to recruit 44 patients. We've recruited 28 so far, so it's going well. And hopefully we will get an idea of whether we can replicate this finding that Gary found in his animal model in man. So I think the conclusions we've got are that, microglial activation does occur in the brain in small cell disease, both sporadic and monogenic. But whether it predicts future disease progression and whether it could be therapeutically switched off really remains to be determined. But if it does, it offers a potential therapeutic target. And I'd just like to thank all the people involved with this. Obviously, a lot of people have been. Stroke Research Group, our imaging centre, which is absolutely crucial. And also increasingly, we're working with Ziad Mallet and our cardiovascular medicine group to characterise the changes in the blood in more detail. John and Audrey in the Department of Psychiatry, and of course, Gary, whose ideas really led to this work. Thank you very much.
Thanks, Hugh. That's great. So there's a question in the chat which you can look at and maybe address in the discussion, but I think we should move on to the next talk now. Thanks for keeping to time.
Yes, we'd go on with another presentation, I would say in a multidisciplinary VASCOG spirit given by Professor Niels Riksen. Niels Riksen is a professor and head of the Division of Vascular Medicine here at the Radboud UMC in Nijmegen, and he bridges preclinical and clinical research by investigating mechanisms of immune cell reprogramming and how this translates to atherosclerosis and also to manifest cardiovascular disease. And also lately he's been involved in expanding this knowledge into the area of small vessel disease. And I'm glad he's willing to talk about the subject in his presentation entitled Reprogramming of the Peripheral Immune System in Cerebral Small Vessel Disease. Niels.
Yes, thanks, Frank, for this kind introduction. And thanks for the invitation to present our only very preliminary work on peripheral immune cell reprogramming. So let me see if I can move forward. No, I cannot.
You've sent the presentation, right, Niels? So I simply request them to move forward the slides.
I'm an internal medicine physician specialized in atherosclerotic cardiovascular disease. And then in this field, really the two main challenges are to improve individual risk prediction, who's going to get a myocardial infarction or a stroke and who isn't, and to improve preventive pharmacotherapy. Can I have the next slide? So I'm a bit reluctant to tell you that these challenges are, in my view, no less in the field of small vessel disease, because you will know that much better than I do. So you can see here on the left that there is a large inter-individual variability in the rate of progression of small vessel disease, which is illustrated here in a large cohort of patients from Frank Eric, who had multiple MRI scans in various years. And we cannot predict yet who is going to show progression of small vessel disease, for example, towards dementia, and who isn't. With regard to treatment, the only thing we can currently do is blood pressure lowering, but otherwise no treatments have been shown to be able to prevent progression to dementia. Next slide, please. Yes. So in this presentation, I hope to convince you that circulating monocytes could potentially be involved in the pathophysiology of small vessel disease and its progression, and as such might provide an interesting target for pharmacotherapy in the future. Next slide, please. So why did we choose to focus on the monocytes? Well, first, a very recent systematic review that you already alluded to concluded that systemic inflammation is probably involved in the pathophysiology. So this is a quote from that paper, demonstrated an elevated level of systemic inflammatory markers at baseline predicted subsequent small vessel disease severity and progression. A second reason is that there are various shared risk factors between small vessel disease and atherosclerotic cardiovascular disease, including old age, hypertension, smoking, and potentially also diabetes. And that we know very well that in atherosclerosis, monocyte-derived macrophages are the most abundant immune cells in these plaques and really orchestrate the initiation, the progression, and the destabilization of atherosclerotic plaques. Next slide, please. So in these plaques, monocyte-derived macrophages are exposed to various DAMs, the disease-associated molecular patterns, so that they will produce cytokines and chemokines, which can subsequently fuel inflammation in the plaque. Also, they can take up oxidized lipoproteins, oxidized LDL, for example, and become foam cells. But what is interesting is that also monocytes that have not yet penetrated the vascular walls, or circulating monocytes, and even their progenitors in the bone marrow, are reprogrammed towards a pro-inflammatory phenotype in patients with cardiovascular disease. And in the next two slides, I will show you two examples from our own group. So in this study, a couple of years ago, we took blood and isolated the PBMCs and the monocytes from patients with symptomatic coronary heart disease, 20 patients and 20 controls. We isolated these cells and stimulated these cells ex vivo with LPS, which is a toll-like receptor 4 agonist, for 24 hours, and then measured cytokine production. And you can see that the cells that were taken from the patients with cardiovascular disease had a higher cytokine production capacity. And on the next slide, you can see that in this same cohort of patients, we also isolated bone marrow. We did bone marrow aspirates, and we isolated the progenitor cells of these circulating monocytes, and also exposed these cells to these inflammatory stimuli. And also these progenitor cells had a higher cytokine production capacity in the patients compared to the controls. Next slide, please. So now, to investigate the innate immune system in patients with small vessel disease, we made use of the RUN-DMC cohort, a cohort that was performed by Frank Galileo a couple of years ago, started in 2006, so 503 patients aged between 50 and 85 years old with MRI signs of small vessel disease. These patients had a follow-up MRI in 2015, and recently they entered the RUN-DMC Intense study, which was a study aiming to investigate the cause of small vessel disease progression in patients without known risk factors for small vessel disease. So patients were excluded when they had carotid plaques and when they had atrial fibrillation. So they ended up with a smaller group of 54 individuals who had 10 consecutive MRI scans 1 month apart to see incident lesions of small vessel disease. And on the next slide, you can see that in the first study that was a retrospective study in this group, we took blood from these patients and related all of these circulating inflammatory parameters, not only to the white matter hyperintensity volume on the MRIs in 2006 and 15, but also the progression between these two MRI scans. And so what did we measure? We measured circulating inflammatory markers, which I will show you in a while. We isolated the PBMCs and performed flow cytometry to look at the three typical subsets of monocytes, the classical, the intermediate, and the non-classical monocytes. And we also stimulated the cells for 24 hours with various stimuli, for example, LPS, to measure cytokine production capacity. And on the next slide, you can see the baseline characteristics. So it's quite an old group, 70 years of age on average. Almost everyone's got hypertension, And you can see on the last row that on average there was a progression of white matter hyperintensities in these 10 years of 5 milliliters. On the next slide, you can see the association between various circulating inflammatory markers, also the ones that you presented before in his overview, like high sensitive CRP and IL-6, and its association with the progression of small vessel disease. And you can see that for circulating IL-6, we could observe a significant correlation between circulating IL-6 levels and not only the MRI findings at 2006 and 15, but also the progression in these 10 years, actually confirming previous large epidemiological studies on circulating IL-6. Then we looked at the monocyte subsets using flow cytometry. And interestingly, we could show that the intermediate monocyte subset, which is quite a small subset in terms of percentage of circulating monocytes, but a very interesting subset because it has a high cytokine production capacity and is regarded as an inflammatory subtype, that these intermediate monocytes also were significantly associated with the progression of these white matter hyperintensities between 2006 and 2015. And finally, we looked at cytokine production capacity. So we stimulated the cells, measured cytokine production, and you can see that we measured 6 cytokines and stimulated the cells with various stimuli like LPS, PAM3-CIS, which is a toll-like receptor 2 agonist, and also Candida albicans. And we measured several cytokines. We observed a significant association between IL-17, which is a cytokine that we also know is important in the progression of atherosclerosis. And we observed a strong negative correlation between interferon gamma, which actually comes from the adaptive immune system. And I will come to that in a bit. We also looked at a subgroup of patients in which we excluded patients with inflammatory conditions like rheumatoid arthritis or patients that used immunomodulating drugs. And in that subgroup, there was also a significant correlation between the IL-6 production after stimulation with PAM3-CIS and small vessel disease production. So subsequently, we did a prospective follow-up study. So the previous study was a retrospective design, and now we use the same cohort to also prospectively look at the correlation between these same markers that I just showed you and the incident small vessel disease lesion in these 10 MRIs. And on the next slide, you can see in the left upper corner that in this small subgroup of 35 participants, so we excluded all these patients with inflammatory conditions, so patients with diabetes, autoimmune disease, or using immunomodulatory drugs. 13 patients showed progression based on the on the white matter hyperintensity, and in 7 of those patients there were incident lesions of small vessel disease. And in the table you can see the baseline characteristics. So the patients with the incident lesions on the right, they are older, they have a higher blood pressure, a lower HDL cholesterol. And on the next slide, you can see, again, the correlation with circulating markers of inflammation. Of course, these are very small numbers. Again, 7 patients only with incident lesions. So we didn't see the same association for high sensitive IL-6, but we did observe that circulating E-selectin, a marker of endothelial cell activation, was higher in this group of patients compared to the controls that didn't show any progression. On the next slide, you can see that there were differences in cytokine production capacity in the 7 patients. So those are the 7 black dots compared to the open dots, which are the controls, for not only IL-6 production after PAM3-CIS stimulation, that's on the right upper panel, confirming the findings we did in the previous study, but also for LPS-induced IL-1 beta production, IL-1 beta also being a very important cytokine in atherosclerotic cardiovascular disease. Then we went further and did a very exploratory analysis, doing some more deep phenotyping of these monocytes. And we isolated the monocytes from these subjects, performed RNA sequencing on them, and compared them to matched patients, matched on the baseline characteristics, who did not show any progression. And here you can see a volcano plot on the left with the differentially upregulated and downregulated genes. And what we observed is an upregulation of several genes that indeed had some associations with inflammatory processes, like EGR2, which is important in macrophage function. Two of the genes important in regulating endothelial function and endothelial leukocyte interaction, and fatty acid binding protein 4, which is important for macrophage function and is also associated with atherosclerosis. And again, we showed a downregulation of the adaptive immune interference signaling, which we also observed in our previous study. So going back actually to the field of atherosclerotic cardiovascular disease, what is interesting is that we know from a previous study a couple of years ago that activation of circulating innate immune cells can also be induced by cerebral ischemia. And this was an animal model of large territorial infarctions, but they observed that very rapidly after stroke in this model, there was an activation of circulating innate immune cells by an increased noradrenaline concentration in the bone marrow niche and subsequently increased beta-3 adrenergic receptor signaling. Although again, this is only shown for large territorial infarctions, I think that based on this, we now propose, and that's on the next slide, that circulating innate immune cells might play a role in some form of some self-perpetuating cycle. So we have shown in our preliminary publications, and of course I appreciate that it's just a very small sample size, but that these monocytes, when they are activated by several inflammatory triggers, can play a role in the initiation and progression of small vessel disease, and that also the cerebral ischemia that is the consequence of that can subsequently also trigger further immune cell activation. So of course the data are preliminary, small groups, but I think it's definitely important to further explore this potential mechanism. So I would like to thank various collaborators. Marlies Nos was the PhD student who performed these studies. Of course, Frank-Erik de Leeuw and his group, and the Dutch Heart Foundation for its funding. Thank you.
Thanks, Niels, for this very comprehensive overview on a topic with which not all of us are very familiar with, I think. So thanks again. I think we're starting to unravel immunology and inflammation and the role it may play in small vessel disease. So thank you for that. And I hope you do have time to stay with us a couple of minutes to join the discussion after the next two presentations. The next presentation will be given by Malin Overmars, if I pronounce your first name correctly. Seems very Dutch, but I've never seen a surname Malin. And you will be talking about a cluster of blood-based biomarkers reflecting extracellular matrix organization, inflammation, and signal transduction relates to cerebral blood flow in patients with cardiovascular disease. Please,
yes.
Malin.
Thanks a lot. So first of all, hello everyone. Thank you for the invitation to give a talk. In the upcoming minutes, I would like to present on our research on a blood-based biomarker cluster that is related to cerebral blood flow in patients with cardiovascular disease. Next, please. Yeah, so first, a short introduction. Cardiovascular disease can negatively impact the brain, among others, through decreased cerebral blood flow. And this may contribute to vascular cognitive impairment. Decreased CBF has been attributed to oxidative stress, inflammation, and endothelial dysfunction. Next, please. But exact underlying processes of cerebral blood flow are largely unknown. Next, please. So we hypothesized that the identification of clusters of blood-based biomarkers that are related to variation in CBF in patients with vascular disease may provide leads on the biological processes that are involved. So a little bit more on our study group from the heart-brain connection study. We had 428 participants with mean age of 67.2. 37% was female and 74% had a history of cardiovascular disease. Next, please. Yeah, and of these patients, we had 92 protein blood-based biomarkers from the Olink cardiovascular 3 panel. And we also had the partial volume corrected whole brain normal appearing gray matter CBF measured on 3 Tesla pseudo-continuous material spin labeling, which looks like this in our patient groups. Yeah, so a little bit more on our approach. So first of all, we looked at individual associations between the 92 biomarkers and CBF. We did that with correlation analyses, and we, of course, corrected for multiple testing. Next, please. And what we found is that 9 biomarkers correlated significantly with decreased CBF. The Pearson's R's were negatives, which means that a higher protein value corresponds to lower CBF. And what we did next was cluster analyses. So for that, we used a string database, which is based on prior knowledge. So for example, from pathway databases or lab experiments. And we also used cluster algorithms with bootstrapping. And what we did is we determined the overlap between the 2 cluster methods. And what we found is that the 9 biomarkers we found before formed one cluster that was associated with decreased CBF, with a Pearson's R of minus 0.23. And this was independent of age. So the third step was that we did a biological interpretation of the cluster of 9 biomarkers. We used the Reactome pathway database for that. And what we found is that the biomarkers from the cluster reflected extracellular matrix organization, inflammation, and signal transduction processes. So that was our approach. And then I already come to the conclusion that we identified a cluster of blood-based biomarkers that is negatively related to CBF in patients with cardiovascular disease. And the proteins in the cluster reflect extracellular matrix organization, inflammation, and signal transduction processes, suggesting involvement of these biological processes in the pathophysiology of reduced CBF. And if this is validated in future studies, these processes might be therapeutic targets for preserving CBF and improve cognitive outcomes. So I can imagine that, yeah, this was a bit quick. So if you have any questions on our methods, on our results, please send me an email and I'm happy to help. And one acknowledgement slide is left, I think. Yeah, so a big thank you to all these people for their time and efforts they spend into this project. Thanks for your attention.
Great, thanks very much, Malin, for that. And we'll move now to our last presentation from Dan Tozer, and then we'll have some discussion. And there's already been a lot of questions about cause and effect. I'm sure this will come back, but Dan is a postdoc in Hugh Marcus's group, and he already sort of set up the presentation that was prospective. So Dan, we look forward to hearing whether PK1195 binding predicts lesion growth at 1 year.
Thank you very much. So we'll move directly on to the next slide as John's already read out the title, please. Just to note, this work is funded by the MRC. Next slide, please. So as you mentioned, or everyone's mentioned really, SVD is a major cause of stroke and vascular cognitive impairment or dementia, and there are no real treatments and only limited understanding of the disease progression. But as Hugh mentioned earlier, this pathway of inflammatory damage leading to blood-brain barrier permeability, leading to increased white matter hyperintensities has been proposed. And in the work that Hugh showed, we've shown that there are hotspots of inflammation as measured with PK11195 seen in SVD subjects compared to controls. And although there was a slight global increase, this was non-significant. Next slide, please. So moving on from that, what we asked was whether these hotspots of inflammation were precursors to tissue damage as measured by progression to white matter hyperintensity at 1 year. So again, the data that we used for this study was MRI at baseline and 1 year with T1 T1 volumes, flare images for lesion marking, and a standard DTI protocol, which we used with FSL to produce FA and then DMAPs. In addition, PET was acquired at baseline with C11 PK11195 over about 75 minutes. And from this, binding potential maps of activated microglia were produced with a white matter reference tissue estimated with a supervised cluster analysis and a correction for vascular binding. Next, please. For this part of the study, there were 19 subjects with sporadic SVD, 14 with CADASIL, and who were a lot younger than the other two groups, and 19 controls. The first step of the analysis was to register the follow-up and baseline flare images together and subtract the lesion masks to produce maps of new lesion voxels. These were dilated and then subjected to a minimum 5 voxel cluster to minimize partial volume effects or slight errors in the lesion marking. In addition, normal appearing white matter maps were obtained from the T1 images using CNX. After this, all the MR and PET images were co-registered to a common space, and the mean microglial activation or binding potential was obtained along with the FA and MD from the new lesion voxels and also the rest of the normal appearing white matter. Next, please. We use this data to answer 2 questions, the first of which is, do voxels which become new lesions at 1 year show increased microglial binding at baseline? And the answer to that was no. As you can see from the graph on the right hand side, in fact the opposite was true, and the new lesion voxels had a much lower binding than the rest of the normal appearing white matter. This was true for the whole population and also for the control and SVD groups, but not significant for the CADASIL group. In addition, no difference was seen between the new lesion voxel binding and the extant lesions for the whole population, but this difference was seen in CADASIL, where the new lesions had a higher binding than the extant lesions for that group only. Next, please. So following on from that slightly surprising result, we asked whether these voxels which become new lesions showed abnormal diffusion characteristics at baseline, and then the answer to that one was yes, for both the FA, which was lower in the new lesion voxels, and the MD, which was higher in the new lesion voxels. There were abnormalities in those voxels which became lesion at 1 year compared to the rest of the normal appearing white matter. When looking at the individual subject groups, this wasn't seen in the control group, but it was seen for the two disease groups. Next, please. So to conclude, then, I think what we've shown is that the lower binding in these new lesion voxels suggests that the damage to the tissue is well established at this point. And so maybe this tissue is less active when it comes to inflammation than other tissue which is in a healthier state. And if indeed inflammation is the precursor to ischemic white matter lesion formation, this process is taking more than 1 year, and we've missed that sort of window of opportunity to study that. This sort of supported by the results that there is diffusion damage in these new lesion voxels, and this is taking a long time and is a relatively slow process. That we didn't see this difference in CADASIL may be due to the smaller number of subjects in this group. It may be due to the lower age of the subjects, meaning they're in a different sort of physiological part of their life to the older subjects, or it may be that CADASIL follows a slightly different disease process compared to sporadic SVD. However, the fact that we did see diffusion damage in these subjects suggests again that there is early damage to this tissue that then becomes lesion. It certainly appears that longer term follow-up is needed to study the time course of SVD in relation to the processes causing lesion formation. Next, please. Just to acknowledge the MRC, the staff at the WIBIC, and other people in the stroke research group for their help in this work. Thank you.
Thanks very much, Dan. Well, very clear, 4 fantastic, clear talks. Now we've got about 15 minutes for discussion. So there were a couple of questions in the chat. Maybe Hugh, could we start with that? Gert-Jan is asking about this sort of primary or secondary process and your thoughts on that. And that touches obviously on Dan's talk as well.
I mean, I think that's such an important question. Is this causal, and if so, where is it occurring in the process? And I just don't think we know at the moment. I mean, I think Dan's data is not what we expected, but is interesting because it does suggest that perhaps this is a much longer sort of cascade and process that we appreciated before. So I don't know. I mean, whether we need to go, whether you need to sort of reverse translate and go and look at this in more detail in animal models where you can look in much higher resolution over multiple time points. I'm not sure, because one of the things also you've got to remember is our spatial resolution is not great with the PET. But I'd be interested in anybody's ideas on this. I think we also are going to need to intervene in the process to really work out what's going on and see whether intervention has different effects of different processes. And the other thing I think is, of course, it's likely we're at different stages in the cascade in different parts of the brain. So I think this is going to be one of the challenges, that some parts of the brain will be quite well established down the pathway to damage, and other parts of the brain will just be starting the damage, which is going to make it even more tricky to look at.
So that links a little bit, Hugh, with the next question from Fatima Jeremiah, which is making a parallel with people with traumatic brain injury. They show axonal injury, and in cases there of traumatic brain injury, there's dynamic activation and migration. And do you think there might be a dynamic process in SVD? In which case,
I think that's a really interesting model to study, because the timing of the insult, and yet we know that after that insult you get progressive damage, which can continue for quite some time. So I think that's a really interesting model to study. I think the difference in our situation is that we've got multiple time points for the damage happening in multiple parts of the brain. So we've not got a single insult. We've got whatever the insult is from environmental and genetic and other risk factors over multiple years. And I think it's going to start in different parts of the brain at different times. So I think it's much more complex than this. But I know there is some similar work in TBI from people like David Sharp, and it will be really interesting to look at the cascade there, I think.
Okay, thank you. We'll come back to the chat. We've got a hand up from Amanpreet.
Hi, I have a bit of a naive question, I guess. How is the tissue damage in white matter different, or are there markers that are different between lesion formations or white matter hyperintensities due to SVD versus multiple sclerosis, for example? Like, I know the pathologies are slightly different, but are there any biomarker indications when we're looking at these tissue damaged areas?
And who's that addressed to primarily?
I guess it was to Dan, the last talk, yeah.
Hi. In short, when you're looking at the fusion damage, more or less everything damages the fusion in the same way. So you're going to get an increase in an increase in MD and a decrease in FA, and that certainly happens in MS and more or less every other disease as well. So the damage to the fusion characteristics of tracts or white matter tissue is not very specific to SVD, unfortunately. It sort of occurs in every pathology. Does that answer your question?
Yes, I was just hoping we can add on an additional aspect that would make it more SVD specific. It was just a curiosity question and maybe a hopeful question.
No, we sort of work the other way and start with the subjects having SVD and look at the damage rather than the other way around. The most of the markers that show damage in SVD, obviously not sort of presence of microbleeds or lacunes, but certainly tissue damage markers occur in MS and other similar diseases as well.
Okay, great, thank you.
I like the interaction we get when people are directly asking their questions to the speakers. I was wondering, Martin, could you address the question you've put in the chat directly to Niels?
Sure. Thanks, Niels, for a great presentation. So very interesting results in your analysis on the immune markers that you showed, including circulating IL-6. Did you control for the presence or the extent of atherosclerosis in known risk factors for athero, given that there is a lot of comorbidity and that it, especially individuals with hypertension and other risk factors, you currently also see an increasing degree of athero?
Yeah, it's a very important question, of course, given the overlap in risk factors. So in the Run-DMC Intense, Frank Eric already excluded patients with at least with a significant carotid artery disease, so with stenosis more than 50%. Furthermore, in the analysis of the cytokines and the circulating markers, we corrected for age, sex, and hypertension. And in the last analysis I showed you, the RNA sequencing, we really matched patients to controls on their cardiovascular risk factors. But of course, that doesn't exclude that somewhere in the body there is atherosclerosis. So it's an important question. We did our best to adjust for that confounder, but certainly important to keep in mind in future studies. Yeah.
Thanks.
Joel Ramirez also had a question to Malin. I do not immediately see you here on my screen whether you're still present here, but yes, Joel.
Yes, Malin, simple question. You mentioned string database analysis. What does that mean exactly?
Well, String is an open access database. It's from the Swiss Institute of Bioinformatics, and it's actually meant to find protein-protein interactions, and then based on several pathway databases such as KEGG, Reactome. Also based on text mining of PubMed articles. So it's like a real nice tool to find interactions between proteins, actually, and based on prior knowledge. So it's very useful.
Thank you.
Yes, thanks for the question.
There's one from Chris Chen. I'll ask it live, Chris.
Sure. Okay. I'm really intrigued by the studies that Hugh and Dan have done along with you, of course, John. So what kind of pathology is really being picked up by these PET ligands in SVD? I mean, I think you mentioned activated microglia, but I think that from the past studies, other cell types also express TPSO, such as astrocytes. How does this link in with what you found? I mean, are these the cells that are being picked up, or are there other cells expressing these receptors, this protein?
You've raised an important question. Dan, do you want to answer it, or?
Not particularly, because it is a very valid point. Yes, I mean, these TPSO markers will pick up anything, will bind to anything that they can bind to. I mean, there's Audrey and John are sort of much more knowledgeable about the specifics of the PET than I am, but there is a certain amount of assumption that what we're seeing is related to microglia activation, and that is related to inflammation. I don't think we can specifically tell you what cellular type it is picking up. Anything that reflects TPSO activation will get bound to by PK.
I mean, I think the binding to microglia is much greater than to other cell types. But for example, they do also bind to endothelium, and we have to control for that in our analysis. So you try and control as much as possible. I mean, this marker has been used for many, many years in other areas. And I think it's the best we've got, it's fair to say. you've always got to be cautious that it's not entirely specific, but it does seem to bind predominantly to microglia within the brain.
But am I correct to say that you didn't find a relationship between the PET binding and systemic inflammatory markers?
No.
You didn't, or you did? Am I wrong?
No. But I mean, we're only measuring certain systemic markers, and I think the relationship between the two is very unclear. I mean, it is very interesting. Yeah. We did find the relationship with BBB permeability, and I was very interested in Valen's talk because I saw you use exactly the same panel that we used. So it'd be very interesting to look across the data, or maybe see whether you can find similar relationships with our data, which we'd be very happy to share to look at. And the sort of the network that you picked up would fit very well with BBB permeability, I think, with increasing BBB permeability that we found.
Sounds good. Yeah.
But yeah, I mean, I think that the key is we don't really know what's driving this process in the brain tissue. I mean, if it's all secondary to intrinsic tissue damage, then you're not going to get relationships with systemic inflammation in the same way.
Aline, would you like to add to this?
Yeah, it sounds good. Yeah, it's maybe we can keep in touch to compare results. Did you also use the Olink panel? Cardiovascular?
Yeah, I was looking at it. I think we used the I'd have to check because it's 3, but I did just look it up, and our one I've got in my slide as number 3.
Okay.
Great, a collaboration form.
The same one you've done.
Raj has a comment. We talked about brain. Raj, you're going to comment?
Yeah, that's a very good question, Chris. And I think with imaging, well, as I said, the limitations have been discussed, but pathologically, we see at least 4 different types of microglia measured and marked by the same marker. For example, the common marker is CD68, which we use for just pathological verification of tissue damage. And we see amyloid, we see sort of really round microglia, same marker, and activated. So the 4 classes, and they vary in range in terms of the burden of vascular lesions that you might have, including, right from lacunes to something that just tissue damage and I'm talking about the white matter. So yeah, I think so. Interpretation of the PK label, I think has to be with caution. I think that's what Chris is saying, and I feel the same.
Well, I think that's true, but as Hugh says, it's kind of the best we've got, and there's a real need for better imaging markers of inflammation. There are some now that are specific for astrocytes. So in theory, you could do studies where you're looking at, an astrocytic versus a more general ligand. I wondered if I could just ask Niels a question, though, because it strikes me in a lot of this discussion, I mean, it's going to be really difficult for us to tease out cause and effect. And with the progression of lesions, there's so many different factors, not least because the size of your white matter lesion to start with is a very strong predictor of how much it changes. So anything that correlates cross-section is likely to correlate longitudinally. And as you were speaking, I did think about all the other kind of patient populations we might be interested in that maybe we're not studying. So people that are having, I don't know, immunosuppressants for other reasons or other things that are interacting with the immune system. Should we be doing more studies of white matter changes in that group to try and get a handle on whether therapy actually does something in terms of stopping lesion progression?
Yeah, that's certainly an interesting idea. When I think about how to investigate possible causality, of course, you can think about studies with an intervention, and the difficulty is that there is not a good animal model for small vessel disease. And of course, in patients, well, the study you do with minocycline is of course very interesting, and it will be interesting to also look at the circulating immune cells in a study like that. Another option is the Mendelian randomization. So there are some genetic variants known to affect cytokine production capacity. So it would be interesting to look at those genetic variations in big cohorts of small vessel disease. But the other way around is what you suggest, and that's also very interesting. Look at patients that are already on immunomodulating drugs and look at the presence of small vessel disease lesions on the MRI. That's certainly an interesting approach. I'm not aware of any study that has done that with specifically this question.
Okay, thanks. I think we'll make this the last question, Martin.
Yeah, very interesting talk also, Dan. Did you consider also looking at the relationship between BBB leakage and incident brain lesions? I mean, having seen your results on BBB leakage and microglia and the divergence of these of the spots, obvious question is whether you did a similar analysis here.
Yes, yeah, yes, to some extent we have and not found a relationship. But what we haven't done yet is split these new lesions, or what I haven't done is split the new lesions into completely new lesions and areas of lesion growth, which may well have slightly different relationships to permeability, and that's something I need to do. But in terms of new lesions as a whole, or new lesion voxels as a whole, there was no relationship. That I say that may be due to lesion growth muddying the waters, if you like.
Mm. And just to expand on the comment from Niels regarding Mendelian randomization, we did such an analysis on various inflammatory cytokines and identified MCP1 as a causal cytokine involved in atherosclerosis. We did not, however, find much evidence that inflammatory cytokines, including IL-6, are implicated in small vessel disease, which of course doesn't exclude that there still is an influence.
Okay, thanks very much, Martin, for that.
I was wondering, TREM2, I was just wondering, TREM2 is coming up as a very interesting marker for a subset of microglia. And maybe is there a radio label or radio tracer for that? Perhaps that's really interesting in the sense that not only it's localized perivascularly, and going with Niels' earlier comments about something from the periphery, something that comes from the bone marrow, might be something that could be looked into the future.
Yeah, I think it'd be great to have there isn't an imaging marker for it. You can measure it in CSF. So there's some very interesting studies that have looked at TREM2 levels in CSF and found associations, but not yet an imaging marker. All right, I think we better keep to time and wrap that up. So on behalf of Frank, Eric, myself, thanks for 4 excellent presentations, a lot of great discussion. Really exciting area, isn't it? I'm sure we'll hear a lot more about inflammation and SV.